Welcome everybody. My name is Jack. I want to thank all of you guys and all my listeners for following my channel and listening to my presentations. Hopefully you guys are enjoying it as much as I am. And if you are and you have not subscribed yet, please go ahead and hit that button on the YouTube channel and follow me on the Facebook page at Side Talks 2.0. Well, today I have a very exciting paper that I want to share with you. And one of the reasons why I picked this paper is that the researchers used a very hot topic in the field of neuroscience called optogenetics to study how visual processing happens in the brain. Optogenetics is essentially the use of light to be able to turn on specific regions of the brain. And now this is a powerful tool that could be used to treat many diseases if we can actually prove its ability to do what it promises to do. So optogenetics is now currently one of the most widely studied fields in the field of neuroscience. Specifically in this paper, the authors use this optogenetic method to answer one of the fundamental questions in the field of neuroscience, which is visual perception. How does it happen? When you see an object in your visual field, where does that processing happen? What are the specific neurons in the brain that are involved in putting together all of this information and giving you this imagery that you see in your head? And to be able to answer this, the techniques of optogenetics is the one way to be able to go around it. So let's take a look at what they did and why this is so exciting. For example, have you ever wondered how or why you actually see the objects when you are going about in your normal day? For example, let's say you're at your favorite museum and you're staring at a statue. Do you ever sometimes wonder, why am I able to see this? Or what is doing the actual seeing? Is it my eyes or is it the brain? Where does all this information go and how does it get all put together? Well, currently our limited understanding of this visual processing is what drives scientists to do these kinds of studies. For now, the way we understand in terms of how this visual processing happens is that we have signals that come from the object, the light actually, that comes from the object. It hits our eye. From there, that information gets processed and travels all the way to the back of the brain in a region called the occipital lobe. The occipital lobe is where we find the primary visual cortex, and it's called the primary visual cortex because that's the first place where the visual information gets traveled into. And from there, we have these networks of neurons, highly densely connected network of neurons, which do a lot of visual processing. And eventually, the net outcome is you get this imagery of the statue in your head. Now, the question is, what are these specific neurons that are involved in, for example, you seeing the statue? Are all the neurons necessary? Are there an assembly or a minor number of neurons that you need? And moreover, here's the more philosophical question. Once we know exactly which neurons are involved in giving you this visual perception of the statue, for example, is it then theoretically possible for us to remove the statue in front of you and instead stimulate these neurons and give you this same perception and mental imagery of the statue? In other words, can we find these specific neurons that are involved in processing this visual picture, in this case the statue, and therefore specifically activate them and allow you to see the statue without having it be in front of you? Now, this sounds like an interesting problem, not only for the field of neuroscience, but from the philosophical point of view. Many philosophers have debated about this topic, and without the advent of optogenetics, it would have been very, very difficult to answer some of these questions. So what is optogenetics and how does it work? Essentially, optogenetics is using light to activate specific regions of the brain. And all you do is shine a wavelength of light, and that turns on neurons, and these neurons that are involved in different kinds of circuits activate these circuitry and give you some kind of a behavioral output. So how does it work? Well, just a really brief background. This exciting tool was developed by Dr. Carl Dijkstra at Stanford University. Here he is shown holding these probes that are involved in generating these bursts of light that activate these neurons. So how does it work? Well, just a quick background of basic neuroscience, and for those of you that don't feel as much comfortable about science or medicine, even if you don't understand these next few minutes of describing of how optogenetics works, you will not lose anything in terms of the main essence of this paper. So please bear with me, and if you can understand it, great. You will have a better, more in-depth understanding of the results. But if you don't, that's totally fine. You should still be able to understand what the overall message of this paper is, which I think is very, very exciting. So how does it work? Inside our brains, we have these cells called neurons, which are the brain cells that are involved in the processing of all the different kinds of information that happens in your brain. So if we look at a neuron, what we see is they have these long wire-like structures. These are called axons. This is where the signal that gets generated from these neurons is able to propagate or travel down and all the way to the tip, from there down to its neighbors. So these are the structures that these neurons use to communicate with one another. So how does optogenetics work? If we zoom in into these axons, what we find here is, let's imagine this being the inside of the axon and this being the outside. Normally, we have these positively charged ions that are sitting on the outside of these axons, and we got these negative charges sitting on the inside. So the way optogenetics works is that we introduce this red protein into these neurons. And what happens is, when we shine this red light, the red light activates this protein, which then changes its configuration, and by changing its shape, it allows for these positively charged ions to come into the cell. And when these positively charged ions come into the cell, they then activate their neighboring area, which causes more positive charges to come in, which then activates its neighboring area and more positive charges to come in. And this is how the electrical signal is able to travel down the axon to its neighbors. So in essence, what we've developed here is a system in which, by shining a specific wavelength of light, we are able to activate a neuron which contains this red protein. So depending on which neurons contain the red protein, those will be the only ones that become activated. So for example, in this case, if this neuron is the only one that has the red protein, it'll be the only one that becomes activated. Whereas its neighbor, for example, this one or this one that doesn't have the red protein, it will not become activated. So this gives us almost a surgical precision in terms of figuring out ways to activate specific neurons and thereby specific circuits in the brain. Now this is really freaking cool and powerful, right? So how did these researchers use this exciting technique to answer one of the fundamental questions in neuroscience? Well, let's imagine here a mouse, and we zoom in to its brain. And inside, let's depict the brain as a circuit board, the same kind of circuit board that you find inside your computer. So all of these connections are different neurons connected to one another. In the back of the brain, just like in humans, we have the occipital lobe, which regulates or which processes information that comes in through the eyes of the mouse. So all of the visual processing begins in the back of the brain, just like in humans. And for the purposes of this presentation, I have drawn here in blue what is supposed to be these neurons that are connected to one another. So what did the researchers first do? First, they showed these animals a visual picture of vertical lines, right? So when the animal sees a vertical line, the information travels from its eyes all the way to the back of the brain, and it activates specific kinds of neurons. And so the researchers first imaged which neurons become activated when the animal sees vertical lines. They then gave these animals a drop of milk. Now I'll tell you why this is important, but just remember, by giving them a drop of milk, the animal associates vertical lines with eating milk. So every time it sees a vertical line, it says, "Where's my milk?" Right? But for now, just remember that fact. And also that when you show vertical lines, specific neurons become activated. In fact, they were able to calculate that remarkably, all you need is only 20 neurons to be able to see vertical lines. They then did the flip. They showed the animal horizontal lines, and we know horizontal lines are not the same thing as vertical lines. So then when they looked into the brain, they saw these green flashing light neurons, essentially different neurons, right? So now they were able to say, well, these ones, these specific neurons that are flashing in green right now, are involved in processing horizontal lines, whereas the prior ones are involved in processing red lines. So then the question now becomes, if we know exactly which neurons are involved in processing vertical lines versus horizontal ones, can we activate these specific neurons and see whether the mouse actually perceives this vertical or horizontal line? This is exactly what they did. So in their next set of experiments, instead of showing the animal a vertical or a horizontal line, they used optogenetics to activate the visual cortex. So first thing that they did, they shined the light onto the occipital lobe and activated lines that are involved in the animal's perception of vertical lines. And the idea is that if we're activating these neurons that are involved in the visual processing of vertical lines, then presumably the animal will be seeing vertical lines in its mind. But how can we know if the animal is actually seeing vertical lines, right? We can't ask the mouse, hey, are you seeing vertical lines when we shine this light and activate these specific neurons? And this is where the key and ingenuity of this experiment comes in. Remember, I told you every time the animal was shown a vertical line, they gave the animal milk, right? So in this case, if the animal is actually seeing vertical lines, then it says, hey, where's my milk? And this is exactly what happened. So when the researchers shined the light and activated specific neurons that are involved in processing vertical lines, the animal started asking for milk. And by that I mean the animal came towards its area where milk is normally deposited. Where when they did the flip experiment, when they activated the neurons that are involved in processing horizontal lines, the animal never came around, because remember, the milk eating behavior is paired with the vertical lines. So what does this tell us overall? It suggests that by shining these light on top of the occipital lobe, we can activate the visual perception of vertical lines in a mouse. Now, isn't that cool? It tells us that we don't really need the eyes to do the seeing. In fact, the seeing is done by the brain itself. Now, this has multiple implications for the field of neuroscience and philosophy. But moreover, we can use these similar kinds of experiments not only to activate visual processing, but other circuitry in the brain. And in fact, scientists have been able to do this exact experiments. For example, they were able to essentially implant or delete memory inside these mice. So what they did was they taught the mouse that every time it moved into a certain area of its cage, it would get a shock. So the animal then associated that area of the cage with a shock, and obviously the shock is a very uncomfortable feeling for the mouse, so it avoided that area. It remembered that that area is related with a very nauseous stimuli. So then what they did was they used optogenetics to specifically turn off the circuitry involved in remembering that that area is not somewhere I gotta be. And when they turned off these signals, the mouse started to venture into those areas. And then when they turned the light off and the memory came back, the mouse avoided that area. So by using this optogenetic method, they essentially created like a light switch where they can turn the memory on or off. Freaking cool, right? In another set of experiments, very similar to what I just described, scientists were able to implant songs into zebrafinch, right? When these animals are born, they don't know how to sing. It takes time for them to learn how to sing. They are taught by their parents how to vocalize and be able to make these beautiful songs. But using this optogenetic methodology and knowing the circuitry involved in producing these songs, the investigators were able to transfer this information from one bird to another. In the field of philosophy and neuroscience, but how is it pertinent to humans, right? Because at the end of the day, does it really help in alleviating pain and suffering or understanding better the human brain? Well, if we go back to our original question, is visual perception happening in the brain or in the eye? We could start off with that. So based on the results presented here, indeed what happens is when somebody is presented even with a blank board, for example, if we know the exact neurons that are involved in visual processing of that same statue we saw earlier, then what we could do is we could activate those neurons and give you that visual perception of that statue. Now you're saying, why is this important? Well, one can think about people who are blind, who can't see through their eyes. And what we could do in these individuals is implant these electrodes into their occipital lobe and therefore activate whatever is in their visual field via these electrodes and allow them to be able to see the world without needing their eyes. Now that's freaking cool. The other cool thing about this is, remember I said we don't only have to activate the visual cortex, we could do other circuitries, right? So for example, we could use the same technology to activate circuits, like in this case shown by flashing green lights, that are involved in depression. And when we activate or turn off or on these circuits, we could actually reverse or alleviate the depressive symptoms in individuals. Or we could activate another group of neurons and circuits that are involved in visual hallucinations or memory. The amount of possibilities is essentially endless. So by doing these very neat set of experiments, Dicerat's group was able to demonstrate that they can specifically activate neurons, and in fact they show that you don't need many neurons in order to get a visual perception. So the hope is that we will be able to transfer this technology into humans, and in fact there are currently genetics technique to help alleviate symptoms of post-traumatic stress disorder. Parkinson's is one of the diseases that optogenetics has the most potential to help. Like I mentioned earlier, depression, mania, schizophrenia, chronic pain, and so on and so forth. So can you guys imagine in the future, you go to your doctor's office, whether you have low back pain or PTSD or depression, and all they have to do is literally shine a beam of light into your head and it would alleviate the symptoms that you're feeling. Now, of course, this is still down the line, but we are not too far away from doing this. Well, this is all I have for today, and hopefully you guys find these to be as exciting as I do, and hopefully you enjoyed this presentation. And once again, I want to thank you guys for listening to me, and hopefully I will be getting some good feedback from all of you, and will have you as my audience again for my next presentation. Once again, thank you everybody, and don't forget to sign up, and have a wonderful day.